actually, she gets her bachelor's degree at this institution too. So,
couldn't get rid of me then. But
this is a time for Anne to show what she has done in the next 4.5 years. Okay, Anne?
okay. All right. Thank you, Dr. Wong.
Yeah, you can turn off the light.
Sure. Hi, I'm Ann Stewart. Most of y'all know me. Have known me for I've been here 8 years. So what we do in our lab, we study chicken genes that contribute to fat deposition. Okay? And basically the reason why, giving you the background, the reason why we do what we do is because obesity is so huge right now. It's not just, it's becoming a major epidemic here in especially industrialized countries like America. Here in America, 2 to 5-year-olds have gone from 5 to 11% in the obese rate, and adults increasing has doubled from 13 to 35, over doubled in just the last less than 30 years. So it is a big problem. We're looking for ways to make the chicken healthier, okay? And that's not the only concern. There is the concern in the poultry industry too. Their major concerns, according to World Poultry, is feeding costs and disease outbreak. What they're majorly worried about in the disease, especially, is skeletal disorders and metabolic diseases. So the safety of poultry product for human consumption as well is very, very important to the concerns of the poultry industry. We have several objectives, and we came up with several theses. We did a whole lot of work on a lot of genes that we tested in the chickens. The first objective were genes that we found to be suspicious on a microarray test where we had raised a group of chickens all on one diet, and we had sacrificed them all at the same age. We took the fattest chickens and the leanest chickens, and we compared them in a microarray, and we got our objective 1. We got our genes for objective 1. And then for our objective 2, we did a lot of research, reading research. Dr. Wong had chosen these 4 because they were so highly publicized. These 4 genes right here, they were highly publicized to have diet effects. Objective 3 was a comparison of housekeeping genes. We wanted to know which housekeeping gene was best for normalization in chicken fat using tests. Can I check this? Okay. And then the last objective, we were to identify SNPs. We know that SNPs contribute to a lot of diseases. So we were wanting to identify SNP sites in the promoter regions of these genes because that, like I said, contributes to a lot of diseases, and that's one of the main concerns of the poultry industry. So these are some of the genes that we got a suspect that they contribute to fat deposition because they came out on our microarray, these and others, as having significant differences when you compare the fat to the lean chickens. Okay? So this is the total group of genes that we have right here that were under suspect, including the 4 that we had read about and got from literature research. And that's what gave us our hypothesis to believe that age and diet could be affecting these genes. Okay? So I'm going to give you a little bit of background of what we know about some of these genes. And that's been publicized, you know, from other studies. IGF-BP2 is insulin-like growth factor binding protein 2. It inhibits IGF-mediated growth and development rates. IGF binding proteins prolong the half-life of the IGF and have been shown to either inhibit or stimulate the growth promoting effects of the IGF on cell culture. They alter the function of IGF with their cell surface receptors. Okay. Gremlin-1's an interesting gene. It's called gremlin because it interferes with cell signaling. It's a cytokine that also is reported to play a role in bone growth, development, and kidney development. It acts as an inhibitor of monocyte chemotaxis. Okay. CCL4, it is a ligand. And it's actually part of the inflammatory macrophage. It actually connects to CCR5. It's the main receptor. You can see here it does a lot in fighting HIV too. It's highly publicized for that. And basically, it's just showing the CCR5 connecting to it. But it is a chemoattractant for natural killer cells, monocytes, and a variety of other immune cells. So, FGFR3, this one it's publicized to be highly conserved between members throughout evolution. I've had one other doctor that I've spoke to that studied this that says he doesn't believe it's highly conserved, but there's still more to be determined about this gene. It's reported to bind acidic and basic fibroblast growth hormone and play an important role in bone development and maintenance. Mutations lead to multiple types of skeletal dysplasia, and that was one of the main concerns of the poultry industry. So this was a gene of important study. EPHB2, it functions in axon guidance during development. It's involved in the guidance of axons that form major connections between the two temporal lobes of the cerebral cortex. And it is reported to play a strong role in tumor growth. So, tenacin C, that's the extracellular matrix protein. It's implicated also in guidance of migrating neurons during development, and it also in neuronal regeneration and promotes neurite outgrowth from neurons. Okay. COL3A1, this is actually a gene that's kind of it's found in the spaces around the cell. It's found in connective tissues like skin, lungs, uterus, intestines, and vascular system. Okay. SCV is a key enzyme in fatty acid metabolism. It is responsible for forming double bonds in stereoCoA. This is how the monosaturated fatty acid, oleic acid, is produced from the saturated fatty acid, stearic acid. SCV is an iron-containing enzyme and catalyzes a rate-limiting step in the synthesis of unsaturated fatty acids. So, okay, and these genes that became of interest was due to Dr. Wong's research. I mean, he reads a lot of papers, and he knows what they're talking about. So he saw these genes as being something of importance to fat deposition. So it wasn't actually one that came up on our microarray results. But GLUT8, PDK4, adiponectin, and adiponectin receptor 1 is actually going to be, it's in my second manuscript that we're hopefully going to get published. So GLUT8. The importance of GLUT8 is to know that GLUT4 is not in chicken. GLUT4 in humans or in mammals is like the most important GLUT transporter. When I remember Dr. Newkirk saying if there is a malfunction in GLUT4, then that's type 1 diabetes for humans. It's actually our main glucose transporter. So, since we know that GLUT4 is lacking in chickens, that they don't have it, we also know that chickens have a very high blood level, glucose blood level. It's twice as high as ours. But others have, talking about GLUT8, others have reported GLUT8 is highly expressed in kidney and adipose tissue, relatively higher than that in other tissues of the chicken. So, PDK4, pyruvate dehydrogenase kinase isosomes, wow, that's a mouthful, encodes a mitochondrial protein with a histidine kinase domains. This protein is located in the matrix of the mitochondria, and it inhibits the pyruvate dehydrogenase complex by phosphorylating one of its subunits, therefore contributing to the regulation of glucose metabolism. Expression of this gene is regulated by glucocorticoids, retinoic acid, and insulin. Adiponectin. This is we know that this is a hormone. Adiponectin is a cytokine hormone originally found to be secreted by white adipose tissue. However, we've now found that it's also in the brown, so the brown adipose tissue, because that's mainly what's in chicken. Others have investigated the expression of adiponectin mRNA in various tissues, and they've reported evidence that it is influenced by food deprivation. So we highly suspected that a diet would have an effect on it, a change in the diet. Adiponectin receptor 1. This is basically the receptor for adiponectin. It belongs to a novel class of membrane receptors that mediate the effects of adiponectin. Others have reported adiponectin receptor 1 mRNA expression is high in skeletal muscle, adipose tissue, followed by kidney, ovary, liver, pituitary, and spleen. So, okay. Coming back to our objectives, the microarray was what first gave us the idea to study these. We also did different sex studies. So this is my son, and he loved the little chickens so much that he named them all. So Mommy had to lie to him in the end and tell him that we took all those pretty little chickens to a farm, and they now live there, and you know they're all at Walden Farms. So don't let anybody know Mommy's a liar. Anyway. To assess the ecology of these genes and their responses to dietary caloric concentration, we took one-day-old broiler chickens that were fed either a low or high-caloric diet from hatch to 8 weeks of age. Okay. The 226 newborn chickens that we got, they were purchased from Ideal Poultry and maintained at Tennessee State University in the Franklin A. Young Poultry Plant farm. They were divided into 24 groups. One half of the birds were kept on a controlled diet of 21 crude protein and 3,040 kilocalories of metabolic energy. Okay? And the other half was kept on a high-fat diet of 23% crude protein and 3,340 metabolic energy. The control diet is basically the low-calorie diet. That is the regular standard diet that is used in the poultry industry. So that's our control. That's the low-calorie diet. Materials and methods. At 1 day of age, the 226 broiler chicks were wing banded and randomly assigned to electrically heated cages. This was just telling you the size of the cages, and whenever they got to be about 4 weeks old, we moved them up to larger cages, and we raised them in the larger cages from like 5 to 8 weeks. Okay? Now, at ages of 2 weeks, 4 weeks, 6 weeks, and 8 weeks, we sacrificed 12 chickens from each diet group. Okay? 6 male and 6 females were taken from the control group, the low-calorie diet, and 6 males and 6 females were taken from the high-caloric diet and sacrificed. And basically what we did is we took out the abdominal fat pocket. They have like a fat pocket, and we pull that fat pocket out. We weigh it, and we also got liver, blood, and pancreas were collected. They were weighed and placed on liquid nitrogen for direct freeze. Both feed and water was administered in a pan until the time of sacrifice. and these chickens were weighed weekly. Okay, this is a breakdown of the diet. Okay, basically what I want you to note about the breakdown of the diet is the low-caloric diet, or the regular standard diet. It had this much corn in the mix, even though it had much less calories. This is the metabolic energy or calorie intake. And the high-caloric diet had less corn, but much more fat in it, and it had more crude protein. Now, the reason why I want you to note that this one has a higher carbohydrate content. And whenever we were studying the GLUT genes, we found some really interesting things on that. That's why we've gone further with that study. But this is the diet breakdown. And let's see. Results overall, the diets worked. It did induce the obesity that we wanted from the high-caloric diet. The overall weights of the chickens that were weighed weekly showed significant difference in chickens during the week of 2 to 7 weeks. So they had significant difference. It was successful at doing what it's supposed to do in inducing obesity. This is a chart showing you their overall body weight. You look at the blue is the low-caloric diet. The red is the high-caloric diet. And during these weeks, we had significant differences. But even in weeks 8 and 1, you could tell that the high-caloric diet was still higher in the fat-fed chicken. Okay. Now, we ran T-test comparisons on you remember the abdominal fat packet that I told you? It has a fat pocket and it's abdominal. We ran T-tests on comparisons of that fatty packet and on the birds throughout the 2, 4, and 6, and 8 weeks that we did the sacrifice. Okay, and the high-caloric diet was always higher. This is actually giving you a graph of it. A high-caloric was always higher, but we had no significant differences between them. But as you can tell, there was always more fatty coming out of the high-caloric diet. Okay, now this is something that I just saw of very interesting, and this is probably something that leads us to hormonal controls, because we know that estrogen does have an impact on fat tissue. The females at 6 weeks of age, when we were doing the sacrifice, from 6 weeks of age to 8 weeks of age, in both diets, the low-caloric diet and the high-caloric diet, from age 6 to 8 weeks, their fat tissue just shot up at this time in the females. Male birds, their bodies tend to be larger. They actually have more body mass on average, or they weigh more on average, but the females seem to be more fatty. So you could honestly say that the males are probably the leaner chicken to eat. Of course, when you're in a grocery store and you're buying a chicken, you don't know if it was male or female or not. So you just buy what you like. The females by 8 weeks of age had slightly over 12.1 grams of adipose tissue than the males. In the high-caloric diet, the females had 12.32 grams of adipose tissue higher than the males. So as a growth pattern, you'll know that the females at this age just really shoot up in fat. Okay, the RT-qPCR results that we did of our genes that we were suspecting, that we got suspected on the microarray that they contribute to fat regulating. We did ANOVAs on those RT-qPCRs. The ANOVA will tell you 3 things. It'll compare the differences among dietary groups, okay, or it'll compare the differences between age, or it will compare the differences by age, by diet interaction, okay? That's what the ANOVA will show you. It'll show you those 3 comparisons. The most significant difference in the gene expression came from age groups that are in genes TNC, EP, HP2, FGFR3, GAPDH, SCD, and ACBD7 and 18F. Those had age group differences, okay? FGFR3, TNC, and COL3A1 showed differences between diets. And differences in the expression of IGFBP2 and GRIM1 didn't have hardly any significant differences at all. FGFR3 displayed differences among age groups, diet groups, as well as by diet interaction. It had the most differences of all, the FGFR3. And as you can see here, the charts show it had a very large P significant difference in FGFR3 for the diet. And you can also see that there were age group differences as well. And EPB2 only had an age difference, and it was basically in the 6 weeks of age. And these two didn't show us any significant differences, but we still reported that. And this manuscript has actually already been written and sent out and accepted. So this COL3A1 had significant differences between diets, and it was mainly in the 8th week. TNC had significant differences between age and diet, and ABC7 and FCD had significant differences in ages only. So basically it was week 8 growth pattern. It was almost kind of like it went kind of up, and then FCD kind of went down. So this is the charting of those differences. What we found about the housekeeping genes that almost every lab uses housekeeping genes. These and there are some others, but we tested these two to see which one was more stable in chicken fat to be the best housekeeping gene for normalization. We found that 18S had less differences in fat tissue, that it would be a better gene to normalize other genes too for lab tests. Okay, going back to the genes that we researched to get suspecting probably that these genes contribute to fat deposition. Okay, we got some really interesting results on these genes, and they're part of my second manuscript. GLUT8 had an extremely high significant difference in dietary group only. Okay, GLUT8 is a glucose transporter. Remember that the chicken is missing GLUT4. Okay, so GLUT8 became extremely interesting because we need to know what transporter is working in chicken fat, because it doesn't have the other one. So remember back to the diet. The low caloric diet had the most carbohydrates, which that would affect why all these had such a major significant difference in diet, so highly affected this GLUT8. Okay, we didn't get any other significant differences in age on this one. It was just the diet effect, but it was a very large significant difference. PDK was a lot like FGFR3. It had all significant differences between age, diet, and age by diet interaction. Okay, and it had very high significant difference. So we know it is affected by age, by diet. And then adiponectin showed some differences among age groups and dietary groups. Now it's been passed that starvation or from other people that food deprivation will cause this gene, will affect this gene. So it did actually affect ours in the dietary group too. But the receptor of it, adiponectin receptor 1, was only affected by age only. It didn't get affected by the diet, nor by diet-age interaction. Okay? Because GLUT8 showed us strong differences, and we know that it's highly publicized that GLUT4 is lacking in chicken, I knew that it had to be, or I hypothesized that it was the carbohydrates in the low caloric diet that was causing it, and I wanted to know what glucose transporter was taking the place of GLUT4 in this species, in the chicken. Well, and I had a high suspicion that it's going to be GLUT8 because it had such a large dietary, it was affected largely by the diet. So, I wanted to know the other major GLUT transporters that were in mammals, and so I wanted to know if GLUT8 could possibly be the one that's actually replacing it. From past studies, we know of 13 GLUT transporters that are in the human. All of the transporters are not expressed in the same tissues. Like what's being expressed a lot in fatty tissue may not be expressed much in liver or pancreas. So I wanted to know what are the glucose transporters expressed in the chicken fat, because such information would be valuable for understanding fat deposition mechanisms. And I use a software called BioRev. We sent off samples to have a transcriptome at Vanderbilt, and in our results I could test what GLUT genes were actually in the fat and what GLUT genes were not in the fat fat cells. So it basically broke down to this. It did have GLUT1, it did have GLUT3. GLUT2 was not found in chicken fat, but it is in the chicken genome. And then the isoforms of GLUT6, GLUT5, it doesn't have the exact same glucose transporters, but it's got isoforms that are similar to the human. And then GLUT7 became really interesting to me because I've never heard it publicized, anything about GLUT7, and I could not find it in the chicken genome. But I've never read a paper out there that's ever told me that GLUT7 is not in the chicken genome. I'm going to go out on a limb and say that I could not find it. It was not there. I could not find it in NCBI genome. But okay. So to summarize and give a conclusion on this, the overall body weight is increased by the high caloric diet, which showed our diet treatment was effective. The RTQ-PCR showed FGFR3, FGFR-BP2, and GRIM1 showed very few significant differences. But ACBD7, EPHD2, adiponectin receptor 1, as well as housekeeping genes 18S and GAPDH showed changes in mRNA expression levels due to age only. So the most differences we got was in age. But age and diet affected expression in TNC, adiponectin, and SCD. PDK4 and FGFR3 showed significant differences among age groups, dietary groups, as well as age by diet interaction. So we had a lot of significant differences in PDK4 and FGFR3. We came to the conclusion that 18S is a more stable housekeeping gene in chicken abdominal fat than what GAPDH is. 18S would be a better gene to normalize to than GAPDH, in chicken fat at least. Chicken abdominal fat tissue is missing GLUT2, okay? But GLUT2 is in the chicken genome, okay? Chicken genome is missing GLUT4 and GLUT7, which is in humans, okay? If you compare the GLUT genes to the human, the main 13, you have main 13 GLUT genes. The chicken is missing these two. And this one is my hypothesis. I came up with that on my own, but it's now a theory because I have evidence to support it. So going on to the other objective, which is the SNP. SNPs, we study these primarily because they contribute to disease on a lot. We took chickens that were all on the same diet. We compared the fattest ones to the leanest ones, and then we compared their gene expression against each other on a microarray test. The genes that had the highest significant differences, FGFR3, GRIM1, IDFBP2, and CCL4, we chose these to study for SNPs because of our evidence on the microarray. Okay. So single nucleotide polymorphism is a DNA sequence where a single position has a different nucleotide between members of the biological species or homologous chromosomes in an individual. SNPs in the gene promoter region can influence whether the gene is expressed or not, okay? And it can cause disease. In order to determine where these expressions differ from DNA sequence, we analyzed the promoter region, and we took about 1,000 base pair area of each of them. Okay. Information on the variations in DNA can cause how organisms develop diseases, how they respond to pathogens, drugs, and vaccines. SNPs are also thought to be key enablers in realizing the concept of personal medicine. You know how you can drive down the road and you see that Vanderbilt sign that says they're custom tailoring their medicines to your DNA. That's kind of what they're going to now. The study of SNPs is also important in crop and livestock breeding programs. As of October 11th, SNP database listed 52 million SNPs in humans. We're trying to seek them out in the chicken. Okay? This is just basically giving you an example of SNP, an AT to a CG. So our thesis hypothesis on this was that there are SNP sites in these promoter regions of these genes, which is could possibly be contributing to the fat effect of these chickens that were all fed the same diet, and still some of them were much, much larger than others. So we examined these for SNPs. And how do you do that? This just goes over how I did it. You would have multiple samples of the organism. Then I designed a primer over the region of interest. I used the NCBI website, the UCSC website, and I extracted DNA. I ran a PCR. I took the agarose gel and extracted the DNA from the agarose gel, and then sequenced the DNA extracted from it, and then I repeated it multiple times and compared the sequence with Chromos and Cat3. So this is just the websites that were handy in doing all that. This one, you can run a blast on it and find your areas of interest. This is Chromos. I'll actually show you the Chromos of FGFR3, and this is also giving you an example of one that we use. You can actually see the SNPs on this one right here, where there's an overlay. You see that this is a a G. The black represents the G, and then the green represents the A. So these double areas right here is showing you the SNP. And this Cat3 example, you had a much larger one that I sent to you in my thesis. This lines up the SNPs in FGFR3, and this is just a short example of it. Okay, and this is our results here. Looking here, we found nine SNP sites in the FGFR3 chromosome, which is showing they had more differences. Kind of like the PCR, it had more differences too, and it also had more differences on the promoter SNP sites. So IGFBP2, this gives the chromosome and so on. It only had 2 SNP sites that we found in it. GRIM1, we only found 2, and CCL4, we only found 1. So FGFR3 looked very promising in finding one that had the most differences. And remember that FGFR3 is reported to be highly conserved, so I I heard another doctor question that once whenever I gave this same this same speech to him. Conclusions for SNP side objectives: we found 9 in FGFR3, 2 in IGFBP2, and 1 SNP in CPL4. We examined the association of these SNPs with fat deposition by detecting linkage disequilibrium, and we found no significant differences between the fat and the lean chicken. What I did is I designed a primer with well, I designed several primers over the areas that had SNPs, and I would place in 2 different kinds of SNPs, and I would know whether or not an A or a T SNP was there. And then whenever we compared them back to how many chickens were lean and how many chickens were fat, we came up about to a 50-50 amount of chickens in both groups. So we basically got no evidence, no significant differences between these groups to show that these particular SNP sites could possibly be some of the morphisms that's causing the weight gain. So this is just going over future work that Shannon here has inherited. We are in the process of new experiments now and completing some. I'm actually working on the second manuscript and we raised several more birds even after these birds were gone. In the fall 2012 semester, there was a purchase of approximately 500 broilers, and I raised those. They were purchased at the same place, but the control diet, they were all fed the control diet, and we sacrificed at 8 weeks of age. So we're actually working on those chickens now. We have the samples of them now. And so objective 3 hypothesis will be future hypothesis will be to associate a fat deposition with SNP markers with a 60K chip for chickens, and we're working on that now. We basically took the fat and the lean, since they were all on the same diet, and comparing them back to each other. Okay, and there's a lot more chickens, 500 chickens in this group. So this is the method of it. And at age 1, the 50 broilers were wing band randomly, and we basically raised them instead of raising them in the metal pens, we raised them in the floor pens. So they were much larger space, but they were all DNA was extracted, and actually Shannon's actually done several cDNA PCRs on them. So that's future ongoing studies that we're doing now. And if anybody's got any questions, be kind and gentle with me. And if you got questions, Dr. Wong can answer them.
From the audience.